Welcome to another episode of Inside UMiami Medicine. I'm your host, Henry Ford, Dean and Chief Academic Officer of the University of Miami Leonard M. Miller School of Medicine. Today, we're going to explore the fascinating world of ophthalmology research and clinical care. From bionic eyes and whole eye transplants to revolutionary gene therapy and the incredible potential of stem cells. To walk us through these exciting advancements, let's welcome to the studio Dr. Eduardo Alfonso, better known as Dr. Eddie Alfonso, Director of the Bascom Palmer Eye Institute, the #1 eye hospital in the nation. Dr. Alfonso also chairs the Department of Ophthalmology at the Miller School of Medicine, and he holds the Kathleen and Stanley J. Glazer Chair. He's a superb surgeon, professor, and researcher, known internationally for his expertise in eye diseases, corneal surgery, as well as ocular microbiology. Dr. Alfonso, Eddie, welcome to the studio.
Thank you, Dean Ford. It's a pleasure.
First, I must congratulate you and your outstanding team at Bascom Palmer for being ranked #1 in ophthalmology for the 21st consecutive year and the 23rd time overall by U.S. News and World Report. How do you do it?
I don't do it. It's a team, as you said.
Yeah.
It's a great group of individuals at the Bascom Palmer Eye Institute that every day put their best effort forward. And they do it with energy, integrity, and compassion. And they don't need to be told what to do. They do it because they think it's the right thing for the patient.
Well, that's fantastic. Well, so while we're on that topic, just give us a brief overview of Bascom Palmer's Eye Institute's position in the field of eye research and clinical care.
We're blessed to have the resources to be able to conduct incredible research and cutting-edge clinical care. And the reason for that is because we have, for example, 80 faculty members. 40 of them are clinician scientists, 40 of them are scientists, and every day they come to work thinking of how they're going to solve a vexing eye disease. We also have other incredible resources in our students, and that to me is one of the most important parts of the Bascom Palmer Eye Institute, that we have the young individuals with the fresh minds, with the great ideas, with the thinking outside the box, that oftentimes they're the ones who come up with potential projects to solve vexing problems. And then we got the resources. Our donor base continues to support a lot of the research, especially the pilot research projects that happen at the Bascom Palmer Eye Institute, that without their support, we wouldn't be able to take the leap and do some of the research projects that we hopefully will talk about.
Well, that sounds clearly like an ideal place to deal with eye diseases. But we're going to get to that because there are so many critical advances that are taking place right there at Bascom Palmer. But before we delve into those fascinating topics, please give the audience just a little flavor for how you became the Director of Bascom Palmer and the Chair of Ophthalmology.
My story is not too different from most of us in the field. We had perhaps an interest in science when we were in high school and in taking care of other people.
But you grew up in Cuba. You were born in Cuba.
I was born in Cuba, and at the age of 7, my family had a little stay here in Miami, but I grew up in Puerto Rico. So from the age of 7.5 to going to college, I went to school in Puerto Rico. I was blessed to go to a school that really had as its motto, meant for others. So we really learned how to take care of not ourselves, but others around us. And then from there, I went to college and medical schools. And during that time, I was very interested in the workings of the brain. And I thought I was going to be perhaps a neuroscientist or a psychologist or a psychiatrist, neurosurgeon, because I was really fascinated by the brain and how the brain supports the mind. And in doing that, one of my mentors said, why don't you look at ophthalmology, since most of the studies you're doing are in the visual system? And it was that mentor who really got me into looking at ophthalmology as a potential career choice. And I found in ophthalmology all my passions in aggregate.
That's fantastic. That's fantastic. It's a kid from Cuba, Miami, Puerto Rico. No, you didn't mention it. Yale undergrad, Yale med school, and now director of Bascom Palmer, and really a great innovator. So, what a pleasure to be talking to you. So let's get into some of the fascinating topic. In particular, whole eye transplantation. Can you explain what a whole eye transplant entails and why it is so important to the field at this point?
The whole eye transplant idea has been around for over 200 years. And the first eye transplants were actually from taking an eye from an animal and putting it in a human being, thinking that that would work. I was talking about 200 years ago, right? But a clever idea. We have been looking at the potential of an eye transplant for 200 years. And more recently, some real advances in the science of eye transplantation have come about. We heard about the unfortunate individual who had a hemifacial, or half of the face, transplanted earlier this year. And by that, we started realizing that we, together with the knowledge base we have in the field, could come up with newer ways to think about the project. So we in the project that's been ongoing at Bascom Palmer for many years of an eye transplant, we now have science that leads not just the technical part of how to do the surgery, but that science also leads the biology on how to get this done. And we're pretty close.
So tell us more about that, because the eye is only one of 4 organs that have yet to be transplanted, along with the ear, the spine, the brain. So tell us, what are the challenges? Why is it so difficult?
Yeah, the main challenge is that when we think of the eye as part of vision, the eye is only a very small part of vision. Most of our vision happens at the level of the brain and the occipital cortex, where there's a lot of processing of the vision that comes in through the eyes. So the eyes are truly a camera that is absorbing a signal and then transmitting that signal to the brain. So the biggest challenge has been, how do we show the brain that this signal that is coming in through a transplanted eye is a signal that it can receive and process? So that's the big challenge. And so we're working through new technologies that exist, especially digital technology, in figuring out how to make that happen. In addition, we have collaborative efforts with not just here at the University of Miami, but with national and international teams on reconnecting the optic nerve. That would be the real way to be able to get that connection going. I think that's going to take a little longer to achieve, but I think being able to digitally connect an image received by the eye to the brain so that the brain can then process vision, I think that is a reality that is close to happening.
So in some instances, choose my rather pedestrian interpretation of what you just said, but in some ways, you're really talking about creating some artificial neural networks so that you can communicate between the eye and the brain. This is the transplanted eye and the brain. And I would imagine that AI should be able to help with that process, don't you think?
Absolutely. Artificial intelligence has brought in a new tool into the tool set of being able to do this, and being able to take that information that comes into the eye and its biological information and converting it into a database that can then be reconverted to a biological information at the level that the brain can read it. That transformation is happening right now in studies that are being done.
Fantastic. So you also have some technical challenges because you have to connect some tiny blood vessels together. And then, as you said, how do you get the nerve to really reconnect?
Sure. That's a very good question, because it is a very miniature environment in which we're working. But the good thing is that there's been technological advances in the instrumentation that is used to be able to look at the blood vessels in the eye, the arteries, the veins, and even the small nerves in the eye. And how do we identify them and reconnect them? And so we've developed a little machine where we can take an eye from a donor and connect it to this machine in a similar way that we today connect the whole body of a human to a machine that can keep that body alive for a number of days. And it's called an ECMO, as is the short term for it. But we are now working with a prototype where we have been able to take an animal eye and keep it alive through a reoxygenation and re-nurturing of the eye in an artificial platform.
So let's break this down, especially for those who are uninitiated. Typically, any organ to be transplanted, whether it's the liver, kidney, or heart, has to be perfused. Mm-hmm. So you're telling me you have developed a machine that can perfuse that eye with the microscopic blood vessels.
Right. Yeah, the reattachment was a challenge because of the size, the miniature size of what we're talking about. But now we have a way to be able to reconnect the main arteries and veins and perfuse through that eye the nutrients it needs to maintain its.
And how long can you maintain that eye?
So that's one of the keys that we're trying to develop right now. That's part of the studies that we're doing, because part of what we want to do is take this eye and modify it. Because maybe your eye, if you donate it to me, maybe your eye already has some defects that I don't want the recipient to receive your defects in your vision. So one of the things we're doing is looking how we can use this eye placed in an artificial chamber and do things to it through science that we already know, through gene therapy, stem cells therapy, plus some technologies, digital technologies that are available to change an eye.
In other words, you are trying to enhance the eye so that you're going to have, so that the recipient will have even a higher functioning eye than the one that was donated.
That's right.
Yeah. Is this like a bionic eye? Because I grew up around the time of Lee Majors and the Bionic Man. Talk about that.
Because of the Lee Majors situation, we don't want to call it a bionic eye. We may have to deal with some legal aspects of calling it a bionic eye. So I call it more a hybrid eye, because it's going to be the combination of a biological eye that we've taken from the donor and doing some changes to it that may be even at the level of what we call hardware. So, for example, the retina that receives the images inside the eye, the retina may cease to be a biological retina and become a retinal chip that receives these images and is able to transmit those signals into the brain.
So you could implant a chip into the eye while it's perfused on the ECMO machine. So it gives me, I'm a pediatric surgeon, so I get to put newborns in ECMO a lot of times, and that's the extracorporeal membrane oxygenator. But hey, just to think about the size of an eye being placed on that device, tell me about the technical challenges. How do you do it? You guys must be phenomenal.
So we have a prototype, and fortunately, that prototype was developed through donor support. But now the federal government has realized that this is a viable way to keep an eye alive using a mini ECMO. So we have just received a grant from ARPA-H, which is an agency of the federal government that gives funding for pilot projects of this kind, to build an additional 4 ECMOs like the one we have in prototype. And we're going to share these additional ECMO devices for the eye with scientists around the United States, so that they can also work with us in a collaborative way in bringing about some of these things that we've talked about.
And you can imagine how powerful a tool that is, because not only can you plant chips and see what happens, and you can manipulate various genes, do gene therapy before implanting the eye. So that becomes a really very, very powerful tool. That was good. So what are some of the challenges relative to whole eye transplant that you're dealing with, given all of the technology and the basic science that you've been able to apply?
Yeah, there are many challenges, and in lay terms, I compare it to planning on our first trip to the moon or our first trip to Mars. That the challenges are significant, but in tackling some of these challenges, we're going to be learning about certain diseases and certain conditions of the eye and the vision that we can then perhaps as tangibles to this be able to address also. So there are challenges. The biggest challenge is, as I mentioned, is going to be that connection between the transplanted eye and the patient's brain. And I think there's going to be a group of neuroscientists that are already working on this, that are going to continue to work on this ECMO-controlled eye to see how that can be done. So what this does is that it brings an in vivo prototype so that we can experiment some of these questions that are vexing as to how we're going to reach the moon.
Now, we always talk about the fact that the eye is immune privileged. So is the barrier just overcoming the technical challenges and some of this in establishing the neural networks, but AI is going to help us solve? Or do we also have to think about how to maintain this concept of immune privilege in the eye? Is that going to be a potential problem?
The immunology or the potential for the recipient's body to reject this device that we're going to be putting in the eye is going to be there. But we know which are the critical tissues in the eye that have more immunogenicity that can really trigger a rejection episode.
What are those issues?
So what we're going to do is actually work on those cells while the eye is in an ECMO device to change its biological characteristics so that when it gets transplanted to the patient, it will look as much as part of that patient as we can. So this is one of the trajectories that we're taking, and I think that will address quite a bit of the immunity issue.
I'm getting chills just thinking about this experiment, this approach, because that is really fascinating. It really is a powerful tool because if you can really reduce the risk of rejection, that's really going to be a true bionic eye, don't you think?
I think it's going to be a great hybrid eye.
Fantastic. We're not going to go back there. So let's talk a little bit about gene therapy for inherited retinal disorders. First of all, what are inherited retinal disorders? How prevalent are these, and why should we be concerned?
Sure. So there are a number of blinding diseases that are inherited, and the most vexing ones are the ones that affect the retina. And about 1 in 4,000 individuals born today will have one of these genetic diseases that will lead to loss of vision. So we already have ways to deal with some of these individual gene-derived diseases. And we have 2 large approaches at Bascom Palmer. Both are named after our donors, the Mark Daly Inherited Retinal Diseases Center, which is here in Miami, and also in Palm Beach, we have the Louis Pope Retinal and Macular Diseases Center. And both of these are looking both at the biology of these inherited retinal diseases and looking at ways to deal with that. That's what we're doing in Miami. And then in Palm Beach, we're running clinical trials that can put into effect some of the bench to bedside type projects.
Let's break it down a little bit. So these inherited retinal diseases or disorders, they're associated with some genetic defects. So it could be, I understand, one mutation, but in some patients you have multiple mutations. Is that correct?
That's correct. And there are 2, or more than 2, but really 2 main ways to tackle these diseases. You can either go for the particular gene so that you can, especially if it's a germline gene, and change it now so that it's changed forever.
How do you change it? Is that using CRISPR gene editing or
Right. Yeah. So there are ways where, for example, for some of the retinal diseases like Stargardt's is one of them, where there is a clinical trial of using CRISPR to change the genes in the retina of that patient, not just, and those genes will be then, in its progeny will hopefully be changed too. But at the same time, there are gene therapies that target function. And this is one of the beauties of vision and the eye, is that what we need to work on is very accessible to us. It's in the eye. So we can go in and do some gene therapies that change how a particular cell functions in producing an element that is necessary for vision. So we haven't changed the cell, we're changing how the cell works.
Behaves. So in fact, you did such a seminal gene therapy in recent years. Tell us a little bit about that.
So we've done really amazing gene therapy work on children that have some of these inherited diseases. And the first one we did had to do with a child who had a congenital disease called Leber's. And we were able to use gene therapy, which was commercially available at that time at a great cost. The company who had developed this used us as the first center to try this. And we were able to treat patients with this condition with improvement in their vision. And today, this continues. So the children that have this particular disease can be treated with stalling the deterioration of the disease, and in some of the patients even improving their disease.
And that's lasting.
Yes, it's lasting. And this was started back in 2017, more or less. Then we also have recently a very interesting observation that one of our clinician scientists made with a child that had a collagen disease, where they form a lot of scar tissue from this particular disease. It's called epidermolysis bullosa. And this child had multiple surgeries of the outside of the eye because it would just keep forming more and more scar tissue in both eyes to the point you couldn't see through the scar tissue. The eyeballs were okay, but the scar tissue in the front was like a dirty windshield all the time to this child.
That's all from the blistering and
the scarring because of this disease of college. So one of our brilliant clinician scientists said there is a particular therapy that is being used for the skin of these children to try to change the genetic makeup of the skin's collagen. And he worked with the scientists who had developed this to develop it into an eye drop so that we could then apply it to the eye. And it was this therapy that has changed the life of this child. And I could go into the science of it, but it's very complex. It's a vector-derived type of gene therapy. But as a proof of concept, what it shows is that things that we're doing to the eye affects the rest of the body, and things that happen in the rest of the body also affects the eye. And we can learn from each other, and this is exciting.
So in that particular case, are you saying that by correcting the problem in the eye, there was also some manifestation of improvement in the rest of the body and the rest of the skin?
Right. So one of the things...
Is vice versa.
Yeah. So some of the things that we've learned is that there is, at the level of the eye, some effect systemically. Yeah. That is very encouraging for other diseases that could have a genetic component, especially in neurological diseases that we know exist, like movement disorders and memory disorders. I think some of these diseases are at the brink of being able to find some newer therapies for them, and the eye is the perfect place to test some of these.
So let me ask you, so what's the number of patients who are affected by inherited retinal disorders, and how much percentage of them have multiple mutations? Because we've talked about great therapy for those maybe with one single mutation. Because it would be nice to know about the rest of the population that they have multiple mutations.
Right.
It's a different challenge.
And this is there's two answers to your question, right? So in known inherited retinal diseases, like I said, it's about 1 in 4,000. But if we look at current diseases of vision and look at what is the genetic potential cause of these diseases, for example, macular degeneration, that I know when I say that word, everyone recognizes macular degeneration because it's so prevalent in the elderly population, and elderly is me, so I'm not calling myself a young Turk. So any one of us after the age of 60 run a risk of developing macular degeneration. Macular degeneration is a multifactorial disease and multi-genetic disease, and it's going to be for diseases like macular degeneration where we're going to be able to find these agnostic gene therapies where we target not just perhaps the causative gene, but also some of the genes that produce the product that leads to macular degeneration.
So you have a pluripotent gene intervention that would affect multiple mutations. So
that's right.
That's powerful.
Yeah.
That's powerful.
Yeah. And like I said, the eye is so easy to access because if we're going to put these gene therapies in the heart, we need to figure out how to get to the heart. If we're going to do it in the brain, we have to figure out how to get into the brain. The eye is so physically accessible, and the retina, that is easier to put. The surgical techniques are easier to perform.
So is there a role for stem cell therapy in some of those retinal disorders or some of the eye diseases in general? Because obviously we have the Interdisciplinary Stem Cell Institute here, and we know that there's been some collaboration with scientists at Bascom Palmer Eye Institute. Tell us about that.
Yeah, so stem cell derived therapies were first done in the eye in the 1970s. And that's because we described...
Targeting which diseases?
These were diseases in the front of the eye. So the limbus, or the circular part around the color part of the eye, on the outside of the eye, is called the limbus. And this limbus has stem cells that grow to form all the surface cells of the eye. And in the 1970s, scientists that I was fortunate to be working with in Boston derived the concept of stem cell-derived diseases of the ocular surface. And back then, we started working on how to replace these diseased stem cells with new stem cells, even from the same patients. It could be that one eye was highly affected, the other eye was not as affected, and we could take stem cells from the healthy eye and put it in the diseased eye. So we learned a significant amount on the biology of stem cell therapy through these early studies. Now today, working with the Interdisciplinary Stem Cell Institute here at the University of Miami, which is a huge resource to all the scientists, not just at Bascom Palmer, but at the university, we have been able to work on not just the surface stem cells, but also on retinal stem cells. And that's extremely exciting because we can look for stem cells that are present in parts of the retina that are not diseased as much as other parts. For example, the front part of the retina seems to be almost a privileged area where diseases don't tend to affect it very much. So we can take pluripotential stem cells from that area and put it in the back of the eye where, for example, macular degeneration takes place, and see those cells differentiate into macular retinal cells. And this is one of the exciting areas that we're working on to try to achieve this.
So it also allows you to grow some artificial, or at least grow some retina in some organoid platform, is that? And you can manipulate it there too, is that right? And do more genetic interventions.
Some of the scientists at Bascom Palmer have developed retina organoids, and what retina organoids are is taking some of these stem cells outside of their biological environment, in this case the eye, putting them in a petri dish and providing them biological instructions on how to develop into a organ like the retina. And this is already happening.
And how does a tool like that help?
It's terrific because now we don't have to try therapies inside the eye when it's in the human, in you. We can try the therapies in the organoid,
In vitro.
see how the organoid is going to behave when we try these therapies.
That's fantastic. So you don't really experiment on the human, on the live individual. So you already can predict what the outcome is going to be before you start. This is simply amazing. So what does the future look like? I mean, for eye transplant, for retinal disorders, and where are we going? And when do you think we're going to have the first successful eye transplant?
Starting from the last question, I think that we will be transplanting an eye in the next 3 to 5 years. And that means that we're going to take one of these eyes that we're currently working with and be able to transplant it to a patient and be able to create some level of connection to the brain, whether it's a biological connection or a digital connection. I feel that it will likely be a digital connection at that point. But I think that eventually we'll get the biological connection going. So I think short term, we're going to be doing that. Is it going to be done widespread? No. I think we're going to be treading very carefully because we have a lot to learn and we don't want to stumble. And so we're going to learn quite a bit from just doing this, but there is a lot of enthusiasm for it. And I think we're going to continue to see in eye and eye research in incredible developments from being able to modify something as simple as me telling you, Dean Ford, why are you wearing glasses? That's something that Ben Franklin came up with, and we're now in a new century. So let's get rid of your glasses, and we're going to be able to do that at an early age. Amblyopia, which is a devastating disease where the eyes don't develop well in children because they can't see, and we can't give them glasses because they live in areas where perhaps they can't get glasses. If we can institute therapies at that very early level to change these defects of nature that happened at some point as a derangement of nature, of evolution, we will be able to change some of these things to a point that is really exciting to me. I think ophthalmology vision care is on for a continued wild ride into the future.
And this magnificent, optimistic outlook, we will bring the program to a close. Dr. Alfonso, it has been fascinating learning all about the great things that are happening at Bascom Palmer Eye Institute under your capable leadership. Once again, congratulations for 21 consecutive years of being rated #1 eye hospital in the world, and 23 years altogether. So great things are happening under your leadership, and we are fortunate to have you as a director and chair of the Department of Ophthalmology.
Thank you, Dean Ford. But more importantly, thank you to the whole group of people that come together to make this happen. I just happen to be one.
Okay. But this has been another edition of Inside UMiami Medicine with the incomparable Dr. Eddie Alfonso. Please tune in for the next edition of Inside UMiami Medicine.